really cool paper I found about a fairly well genetic disorder called mucopolysaccharidosis type 2 and attempts to treat that in fetal mice. Now, to begin with, what in the world is MSP7 or mucosaccharidosis type 2? This is an autosomal recessive disorder that affects the body's ability to get rid of glycosaminoglycans or GAGs. These gags can actually lead to multiple organ damage if they're allowed to accumulate, and oftentimes they lead to something called uh, hydrophytalis, which is what you see right here in red, which is basically fluid builds up in the fetus and it results in it actually being stillborn. So, because of this, the current treatment is after you're born with this condition, if you actually survive, then we give you enzyme replacement therapies or we transplant stem cells to try and actually increase your ability to fight off the different disease phenotypes that come with this disorder. But what this paper hopes to show is that if you use a prenatal intervention, you actually can increase the effectiveness of the treatments. Now, one reason you want to do a fetal prenatal intervention is because MSP7 kills a lot of children or a lot of fetuses before they're actually born. They're stillborn. Another thing is because we're using enzymes that are basically made from recombinant DNA, your body can produce a autoimmune immune attack against it, which requires you to go on immune suppressants to actually receive treatment. But if you produce them as when you put them in when you're a fetus, your body will actually become more tolerant and won't produce the antibodies against the therapy. The third big reason is because it can pass the blood-brain barrier, which therapies after you are born cannot do. And also, because we're intervening earlier, we're decreasing the amount of accumulation, therefore decreasing the actual disease severity. So, experiment one, they took two mice that were heterozygous for the mutation that causes the disease. This mutation, the reason they're not homozygous for the disease is because the homozygous mice often are sterile and can't breed. So we take two heterozygous, we breed them together, we inject either the fetuses with either PBS, which is basically a control, or with the enzyme replacement therapy about 14 days after they're implanted. And then once they're born, they receive a booster of the enzyme replacement therapy to help increase survivability because these mice often die early. Then 8 to 10 weeks later, we do multiple different tests, which we'll go over to see how did this work compared to conventional therapy or no therapy at all, and compared to you being born normal. So, first, what we're looking at is survivorship. So this is survivorship to both. If you received PBS, around 40% survive compared to 50% if you received a treatment. This is more obvious though when you're looking at survivorship to when they're weaning. So in PBS, if you survive, there's a 20% survivorship to weaning with this genetic disease if you're injected with PBS. But with the therapy, it actually goes to around 35%. So we're talking about a major increase, a 15% increase in survivorship to weaning. We also can see from here that the UTL, the enzyme replacement therapy that was used to treat that were injected with the therapy when they were fetuses also had the only positive deviation as far as survivorship. So these both had negative, which means they were below what was expected as far as survivorship goes. So the next thing we have is what basically what they're doing here is something you might be familiar with. It's basically, what's it called? It's basically it's a cell sorting technique where they're sorting out cells based on their fluorescence. And here they have a negative control, and then based on the fluorescence, we can tell whether B cells or T cells are produced. And here again is basically what we're looking at here is something called GUS activity. GUS is the actual enzyme that breaks down these glycosaminoglycans in the body. So increased activity would be a good thing because that means they're breaking it down more, which means there's less accumulation. So, figure 2. These results were attained by taking brain tissue samples, specifically looking at something called microglia like cells. Now, in the paper, they actually call them microglia till like the final paragraph when they said we should call them something else. So, these results are important because we won't see if this actually does anything for for actually training the brain. Because training the brain with this is always really difficult because you actually have to inject it into the brain if you give if you give neonatal and postnatal therapies. But the whole goal of this is that prenatal will be able to actually pass the blood-brain barrier because the blood-brain barrier isn't really formed yet. And what we see here is that the treated cells have an increased count. What we basically what you should see here is that GUS activity is increasing when you have injected mice compared to the non-injected. Now, the reason these two are so high are these are normal phenotypes because this is a plus plus mouse, which means it has fully normal genes. This is a plus minus. This is a heterozygous, but it's still normal. This is one that has the disorder, but it received the injected therapy, and we see increased GUS activity in the brain compared to both the minus minus and the therapy as an adult. Even that doesn't produce a lot of activity in the brain. So we next move on to, again, more this type of information about the brain. And again, we see a lot of important GUS activity related things where why GUS is not increased nearly to the normal standards, it still increased a lot compared to the control having the disease phenotype. We also see, and this is something I want to bring out here because this is a picture and therefore it's a little nicer and a little easier to look at. This was something they did where they screened, where they were basically looking to see how much accumulated glycosaminoglycans were. So here you see that in water type, we don't have that much accumulation. There's no dye. The untreated, you have lots of these dark circles, which is the accumulated glycosaminoglycans. And in the treated, it's nowhere near the wild type, obviously, but it's definitely lighter than the untreated, which is a very qualitative way of saying it's working. It's not working as well as the wild type, but it's definitely working in this very difficult thing to treat. So another thing that was tested was grip strength because, of course, the mice, one of the things that are affected is both skeletal and grip and muscular strength in mice that have this disease or in humans who have this disease. So this is the normal grip strength with no treatment. As you can see, the grip strength decreases when it was shown when you have the disease and no treatment, and with the treatment with the disease, your grip strength actually increases, seems relatively similar to the normal grip strength, which is both shown in that it's statistically significant, statistically different compared to the non-treatment, which you can see by this little asterisk up here, that's what the asterisk means, but they're also not significantly different between the treatment and the wild type phenotype. So both of these have very similar grip strength, which is very good if you want to make this into a potential therapy. This is evidence you can provide saying, hey, my therapy works in mice. Now, as with Figure 2, this is providing information. Figure 2 provide a lot of brain and neurological stuff. Figure 3 is going to provide our organs, our tissue samples. Oh, before I continue, I should probably warn you, there are 8 figures in this. So I'm sorry if this is a little long and a little rambly. If you have any questions, please post them in the comment section. I'll answer them at the end. Again, I'm really sorry. I should have said that earlier. So with this, again, we're looking to see the accumulation of those glycosaminoglycosides, those GAGs. And again, we see in the untreated, this is represented by these big white spaces in both the liver, the spleen, and the kidney, less so in the kidney, but in the liver and the spleen, definitely, you can see this big accumulation of sources. So, you can see this big acclimation of these aminoglycans, these large white spaces, which is non-existent in the wild type. And again, the cool thing is that in the treatment, while you can definitely see there's some very small ones. I mean, the spleen is really gets heavy in this, but what you can see is that they're definitely smaller and they definitely decrease, which again is a great thing for this being used as a potential therapy or going on to clinical trials. And another thing that is often affected is the actual bones. So a normal heterozygous bone, female bone, for this. Okay, that's actually a really good question. Uh, Before I go on about the bones, I'm going to explain why we care about glycosaminoglycans. So they do play a role in signaling. That is actually a good thing to bring out. They play a role in signaling. They do several other things as well. But the reason that they're dangerous when they accumulate is because they're basically they're dangerous, kind of like many times your body doesn't know what to do with them, so it basically stores them, and this over time, this storage of them causes damage to the tissues they're stored in. So your body needs these glycosaminoglycans for several things, but as soon as it needs done using them, it has an enzyme come along and breaks them apart. uh so uh Basically, that's why we do the enzyme replacement therapy, is we're trying to basically put back in this enzyme that goes and breaks apart these molecules. But they are important for signaling and other things, but as soon as they're used for whatever they're used for, your body quickly breaks them down because it can't let them accumulate, or they're toxic to the cell in high volume. And basically, these people just can't get rid of them. So I think that's a good explanation. Please tell me if I've made anyone more confused. Thanks. Uh, no. Anyway. Okay. Anyway, one thing that is affected is bone length. Femur length is what's measured here because the femur is a really large bone, which makes it easy to look at. What you can see right here is that the normal heterozygous, which were what the parents were, this is a normal femur. And with the lack of any type of treatment, the femur majorly decreases in size, which can be seen. This is what these graphs are actually representing. Except one graph is representing the tibia, which they don't actually have pictures for. But C is representing directly in graphical form what is being represented here in picture form. So with the treatment, as you can see here, we get this statistical significance difference between the actual I don't receive any treatment and I receive treatment. My bone actually grows longer, but it's again, this is one of those things where it does grow longer, but it's still not nearly as long as the wild type. But still, we're decreasing the actual disease, the severity of the disease, which is the whole goal of this treatment in general. Not so much to cure the disorder, but to lessen the severity of symptoms. So back to this. This is going to look very similar to what we've seen a lot of, which is we take the two mice, we breed them, they make babies, we inject the babies, and then 8 weeks later, we check something. Except in this case, before they were using the actual native treatment, the native protein for these mice. This is all about studying that whole idea of if you inject something into a fetus, it will not develop an immune response to it. Idea that it will become tolerant of the actual treatment. And so what they're using is something called human recombinant GUS or glucosidase, which is again the whole enzyme, a version of the enzyme that breaks down these gags. So again, very similar, but this whole idea is not testing does it work. This is testing, well, do we actually see increased tolerance for treatment? And so what we're looking at here is antibodies, IG1 and IG3. And if I give the treatment to a normal, non disease mouse, if you see here, I get a high amount of antibodies. Actually, no, scratch that. These are all disease mice. And without the in neuro treatment and with just me giving you basically the therapy, we get a high amount of antibodies. Your body sees this as or the mice bodies in this case, see it as a foreign invader and tries to destroy it with antibodies. But compare that to this. This receives the actual in utero therapy or the prenatal therapy, as I've been calling it. You see here, it's really low, like really severely low, which is exactly what we expected, which is what is really good. It says that in utero therapy, not only are you tackling this disease earlier, but you are also increasing the patient's tolerance for the treatment to the point that they all produce almost no antibodies to try and destroy the treatment, which actually is a great thing because it means they don't have to go on immunosuppressants, which if you can imagine trying to put a 1 week or 2-month-old child on immunosuppressants also comes with the risk of children this age already have weak immune systems, and putting them on immunosuppressants only weakens their further, which increases the danger of them getting a deadly disease or deadly infection that they would normally recover from. So this is actually a really cool result. This is normally when we think about medicine, we want really high antibodies. We want to know our body is actually attacking something. In this case, we have really low antibodies, and that's what we're looking for. So, figure 5. So again, mice were bred together, but this time we are doing hematopoietic stem cells. Now, the reason that we're doing hematopoietic stem cells instead of enzyme treatment, replacement therapy, which we have been talking a lot about before now, is because these stem cells will become cells that will actually produce the enzyme naturally. The idea is that this is a longer lasting treatment. This doesn't require you getting a shot or an injection or taking a pill every day. This might actually help, and this is a therapy that is recommended for like postnatal, you're born with this disease, currently available treatments, is that along with enzyme replacement therapy, you get this type of transplant to try and increase the natural amount of gus being produced by your body to break down gag. So in this case, we're seeing, well, if we implant it as an embryonic stage, how much difference does it make? So that's what we're about to see. And again, another big thing here that I'm about to forget to mention is that this is, again, are we going to be able to go past the blood-brain barrier? Which is, again, a big problem with this current treatments, because current treatments have to be injected in the brain to prevent the buildup of gags in the brain. So again, we see brain functional and engraftment studies. We're basically saying, did it go to the brain? And again, we have these flow cytometry stuff. I just remember the name of the cell sorting method. I've been trying to remember this entire presentation. So flow cytometry, we see, again, basic flow cytometry stuff. But what we're also looking for here, which I think is really cool as a geneticist, is we're seeing the percent chimeritism, which is what is right here. So what that is basically saying is how much of different cells, the native cells of the mouse compared to the cells we transplant into the mouse, are actually in the blood. This is called chimerism, where you have cells from two different genetic sources. It can occur naturally. Sometimes twins that are, when you have you will have two twins, fraternal twins, where each one is genetically different, and over the course of the pregnancy, they will come together to form a type of chimera. Of course, that's a little more complicated than what they're doing. But anytime you receive an organ transplant, you now are considered a chimera, because you have your native genome and the cells that contain that, and then you have the transplanted organ that contains a completely different genome. So this is looking at the chimeritism, and how does it lead to, how high of a chimeritism do you need to produce actual results? And again, this is CD4 cell plus cells. Basically what we're looking at, if I read the information correctly, which again, I'm really, this, anyway, we're basically checking to kind of something similar to GUS activity. How well are our cells working at producing a treatment? So figure 6 is more about this stuff, with more about the transplant and the hemorrhagic stem cells. We have, again, this very similar wild type. We have the chimera, and then we have the control. With the control, we have these big white spots, which is where there is an accumulation of the gag molecule. And again, because of this causes the disease. But as we see, the chimera and the control have a decreased amount of these giant white spaces, which is what we're exactly looking for. So again, this is very these show that it's very similar to the enzyme replacement therapy, that we're actually decreasing the disease phenotype, that you don't have this affirmation, which is a great thing, because this is a more permanent therapy. As I said before, this doesn't have the I have to inject myself every 2 weeks thing. Now we get to talk about something that's pretty cool. We get to look at a dissected brain, which is it's kind of early for Halloween, but here is your mad scientist moment you can have for the day. Basically, what they're looking at is these things they're showing is where the cells have actually migrated to the brain. These cells that are producing, because these cells are actually producing the Gus molecules. Now, to explain why something would might how hematopoietic stem cells, so stem cells that come from the blood, why are they in the brain? Microganglia cells, if you don't know this, Gailey cells, are the only immune cells found in the brain, and they come from, all immune cells come from blood stem cells. So basically, we're trying to see, will the stem cells we transplanted in utero be part of the group that goes to the brain? And how high of a chimerism do we need to actually produce a noticeable change in phenotype? This is just proof of concept they actually made it to the brain, which is a big deal. What you have here is a very old technique that I actually didn't think anyone used anymore, called a microarray. And basically, to spell a lot of confusion, because this took me a while to wrap my head around. All they're doing is checking to see if there's any change in gene expression between the transplanted stem cells and the normal actual microglia in the brain. Basically, are they acting differently in any other way besides producing Gus? And the truth is that there is one protein that has significantly less expression, but it does not change anything about the phenotype. So all this big, colorful heat map microarray comes down to is that basically there is no change in the there is no change in the gene expression between the hematopoietic stem cells and the actual native microganglia cells, which is good because, again, we don't want to change too much and accidentally cause another disease while we're trying to cure or treat this one. So, final picture, final thing. Again, we're seeing basically they're doing more of the, we have stem cells here, and we have the, what basically they're doing is they're looking at fluorescence. These highly fluorescent ones are the chimeras. This, what we see here is that the wild types have high fluorescence for these two and very low fluorescence for the, basically what amounts to CD68, but very high red and green fluorescence. The disease has very low green fluorescence, very kind of normal. I mean, to me, these look very similar, but this is supposed to be higher than this one, fluorescence. For the red fluorescent. And then the most obvious is there's a lot of CD68 fluorescence here and none in the wild type. And in the actual chimeric cells, we see, look at this. It's really kind of cool. It looks nearly identical with the CD8 fluorescence to the actual normal disease-free phenotype. I mean, it's less obvious for the wild type green fluorescence. Here, they have a lot of high fluorescence, and here it's like you have a few islands of it. But here you can see a big difference, how very similar they are, and how very different it is from the disease phenotype. So, to sum this all up, let's discuss. What does this all mean? In utero treatments actually do improve several phenotypic markers compared to just giving the treatment after they are born, the mice are born. And it increases Gus activity in the brain. It also decreases antibodies against potential treatments, which means that these treatments are better tolerated. Now, a major limitation that if you read this paper, which I really recommend doing because it's a really great paper, I recommend you will see is that poor survival rate. A lot of stuff is done in this paper to try and increase the survival rate of the mice. Basically, that's why all the mice, if you look at all this, they will see boost, boost, boost. All these boosts are doing is trying to make sure they survive long enough so that we can actually collect data on them. And even then, the sample sizes were much lower than the authors would like. But the really cool part and what I think is a really amazing part of this paper is how it ends. This treatment is actually in stage 1 clinical trials right now. At least according to the authors it is, which is an amazing thing because you don't often get to read a scientific paper and then at the end read, hey, this is actually being right now tested for actual applications in humans. So this was just a really exciting thing. I wanted to kind of let everyone know that this research right here is very like groundbreaking and new, but it also is in the middle of actually being used as an actual practical, being researched as a practical therapy and it's being tested as a practical medical therapy. Anyway, now that you've listened to my long spiel and me talking way too fast about this paper, uh, you can ask me any questions you want. So, do we have any questions?
Hi, John. I think I just missed this in the beginning,
Yeah.
but what is this treatment trying to solve, like trying to fix?
This treatment is trying to fix the acclimation of glycoaminoglycans in tissues in different organs. So what the actual therapy they're researching what is trying to fix is that most fetuses do not actually survive the disorder long enough to actually be born for actual therapy. So they're stillborn. So this whole idea is that we're trying to treat them to prevent them from being stillborn. We're also doing this to prevent them from actually having an immune response against therapies, which requires us to put them on immunosuppressants and stuff. Basically, the reason we're doing all this in utero stuff, even though most of this stuff is proven to work, in neonatal conditions, is because most of these will not survive to be born, and even if they survive, there's several other problems that come with trying to use these therapies on infants. Another big one is that there's concern that if you wait till they're actually born, there's too much acclimation and too much damage have always been done to the cells. And this actually is kind of like oleo, trying to treat it oleo to prevent that acclimation.
So in clinical trials, they're testing this in utero, like before the
Yes,
baby's born.
yes. Which I think is really cool. But
Yeah, no, I think going off John's last point, I think it's really cool that like just the process of publishing a paper takes like years sometimes. And then the fact that by the end of it, they were already able to like go into phase 1 of clinical trials, it's really cool.
and this is actually a really new paper. This was published in 2020. I forget exactly when, but and who knows, the clinical trials might be in the middle of yes. To answer that question very basically, yes, these are always approved therapies to treat people who have this disorder. And this is something like if you were born with the disease, you would have enzyme replacement therapy every day of your life, basically. Or you would have this therapy would be part of your daily routine. So yes, these are always approved therapies. They're just not approved fetal therapies, which most people don't actually know is a thing. I didn't know it was a thing till like 2 years ago.
So how many people are affected with this condition?
It's actually a fairly, according to from what I did a little background, and it's a really rare disorder, but the authors actually think it's a little more common than people think, and that it just so happens that most fetuses that have this disorder are stillborn. So they die before they're actually diagnosed because most of the time they from what I can tell, this is not often caught in just your basic one-up genetic test to see do you have Down syndrome or something like that. Most fetuses will die from this before they're born, and so they think the number is actually higher than what is currently said, but overall it's a well disorder. If you actually go on the actual website of the people who look at this, which is in the which is in the citation thing. So there is something in here about, if I can find it, yeah, about MSP7. This is actually called slide disorder. This is actually a website where they focus on treatment and therapy and providing patients information. It's classified as a rare genetic disorder.
John, do you think that the treatment for MSP7 could be applied to any MSP? Because I understand that enzyme disorders, right, from a genetic.
Yes, they're all lysosomal enzyme disorder. There's a lot of different lysosomal enzyme disorders that are called MPS something. I imagine that actually this could be applied to different ones. Now, I don't know much about the other MPSs. MPS2 I know is really common. I don't know much about how the genetics of how that works, but I imagine you could take this research that's done and this clinical trial being done and then apply it to our disorders, MPS disorders, pretty readily, and probably using these clinical trial results and stuff kind of save yourself and jumping as through many loopholes or jumping through as many hoops to get your trial to the hospital. So yes, to answer your question shortly and straightly and not with as much blabbering, yes, I do.
Do you think in utero treatment should be applied to other enzyme deficiencies, or just perhaps in like areas where there aren't access to proper foods or something? Could an enzymatic treatment be used to ensure people are still receiving proper enzymes to break down certain things?
Okay, so I'm gonna make sure I get your question correctly because I'm hearing this could be completely because I'm not hearing you correctly. But I hear two questions. One, can this be used for our disorders like this, or should it be used for our disorders? And I would say it depends on the severity of disorder. From my understanding, MPS2 doesn't actually present any symptoms till you're much older, in which case it might not be worth the risk of a fetal intervention or in your intervention. And then your second question is, well, can we use this in parts of the world where getting certain nutrients is very difficult to try and increase survivorship of fetuses or prevent stillbirths? Is that correct, or did I completely miss that?
That's
the optimistic scientist part of me says yes, we should. That would be a actually good idea. If you take, there's actually a class on this type of stuff, or very, very loosely this kind of stuff at NC State, but they do it with chicken embryos, and always chicken embryos are injected with nutrients to try and increase their survivorship or increase their weight and stuff. But well, that actually is a practical thing to do where nutrients deficient, where there's areas of nutrients deficiency, where most places with nutrients deficiency lack, high quality medical care, or people who suffer from nutrients deficiencies often lack the money to go to a hospital and have this. The optimist in me says yes, it could be used for stuff like that, but the outside me says that it's why it could be is probably not a very effective way of doing it.
Thank you.
Does that make sense, or am I babbling?
No, that made a lot of sense. That was a fantastic explanation. Thank you.
Thank you. Oh, sorry.
I'm sorry. I was just going to ask to make sure there was no other questions. Okay, then. Thanks for listening to me.
Thank you, John. You made a really complicated topic seem really easy to understand. So up next, we have Trina, and she'll be telling us about her research.
John, could you stop screen sharing for a second?
Oh yes, I'm sorry.
No, you're good. You're good. Okay, you guys should be seeing a blank white screen.
Yep.
Are you seeing that?
Yeah.
Okay, I'll get started really quick. Hi everyone, I'm Trina. Thanks so much for sitting and listening to John and I today. So what I'll be presenting on is part of a project I did for honors credit in a genetics course this past fall. And so when I was assigned this project, I was told to choose a topic that was interesting to me, which is very broad, but I decided to take that opportunity to step outside of my comfort zone and to choose something that is actually not one of my research interests, and that is the common cold. And the reason why I'm interested in it a little bit is because I've been told all of my life that there's no cure for the common cold and you just have to get sick and get over it. So when I first started researching this and checking out news articles on it, I came up with this first one, which is from the Washington Post. And pictured is Dr. Jan Corrett, and this is a really interesting paper, which I have loaded in the drive in the folder with my name if you're interested in reading that. But another one that I came up with is this one from the Imperial College in London, England, and that's what I'll be focusing on today. But before I get into that paper, I'd first like to discuss why a cure for the common cold doesn't exist yet. So there are approximately 160 strains of rhinovirus that can cause the common cold. And the rhinovirus is just a major player in it, and there are lots of other viruses that contribute to this condition. There's also the fact that viruses mutate extremely fast, and so fast that scientists simply can't come up with vaccines or cures fast enough to match the rate of mutations. And to compare the situation of the common cold to the flu, scientists only have to target 3 or 4 strains of influenza per year. So it's definitely a much more approachable problem when it comes to the flu compared to the common cold. And I'm sure that when you first heard me talk about the cure for the common cold, you probably thought, why does there even need to be a cure? Can't everyone just get sick and get over it? That is true, but it's extremely inconvenient. Some people miss only a day of class or work. Other people miss a week or a week and a half. And there's also the fact that we don't even know if it gives us any meaningful immunity, because people do get sick with the common cold multiple times throughout their lives, and even multiple times within a single year. And although the common cold isn't lethal for most people, it is for those who are immunocompromised. And these people include people with asthma, COPD, or cystic fibrosis, as well as other conditions. And even more, some strains of enteroviruses, which is the overall family that rhinoviruses fall under, can cause severe symptoms like paralysis. And of course, that can be deadly if you're not in a hospital setting, especially. But back to the paper that I was talking about. Here's a picture of that, and it's titled Fragment-Derived Inhibitors of Human N-Myristoyl Transferase Block Capsid Assembly and Replication of the Common Cold Virus. Now, that's a lot of words, but I'll attempt to break this down. But first, I want to start with viral replication overall. So when viruses replicate, there's the host cell, and here's the virus. They go on to inject their genetic material into the host cell, and typically that's RNA. Then the host cell begins to translate that mRNA into viral proteins. And at that point, there are more modifications made in order to produce more progeny viruses. That's viral replication overall. But more specifically, with rhinoviruses that I was talking about earlier, there's a co-translational modification called myristoylation. And that occurs when the host cell enzyme called N-myristoyl transferase, or NMT, takes a fatty acid called myristate and attaches that onto a part of a protein called the VPO. Now, once that VPO is myristated, that can go on to form a functional protein. But more specifically, that functional protein plays a crucial role in allowing progeny viruses to have a developed capsid or outer shell. So without this protein, they can't survive outside of the host cell. Now, in the paper, the group of scientists wanted to target this specific mechanism and stop it. And they did that by coming up with an inhibitor called EMP1088. And EMP1088 is something they synthesized, but is derived from a naturally occurring inhibitor called EMP72 that occurs in the human malaria parasite. The reason why they chose that inhibitor to derive their new chemical off of is because the region I have circled, the dimethylamine, interacts with a very specific tyrosine residue that's conserved in human NMT. And I have both EMP72 and EMP1088 pictured there. So because this mechanism is targeting human NMT, it shows great promise theoretically. And so once they synthesized this compound, they went on to test it. Now this figure that I have displayed is observing the cytopathic effect in cells. Now the cytopathic effect it describes structural changes that occur in host cells after a viral invasion. So what they did here was they took HeLa cells and infected them with a strain of rhinovirus for 2 days. And then they looked at these cells to see how much of the cytopathic effect had taken place. And what I would like you to gather from this figure is that less of the cytopathic effect is observed in cells with EMP1088 compared to EMP1031. And EMP1031 is a control inhibitor that they already have baseline values for in terms of the effect. Sorry, my thing stopped clicking. Now this next graph I have is observing viral counts in HeLa cells again after they've been infected with multiple rhinovirus strains, but with the 4 strains that I have displayed. So the cells were infected for 6 hours with the strain that I have, and then they were treated with either DMSO, which is a control, or their experimental inhibitor, EMP1088. And what I want you to gather from this figure is that there were significantly lower viral counts in EMP1088 treated cells in all 4 strains. And so we saw the cytopathic effect, and we're seeing this, which both are great. They're completely supporting that EMP1088 is working well. But if you look at this next graph, this shows that HeLa cells, so in here, HeLa cells were infected with a rhinovirus strain for 6 hours. And then EMP1088 was added at different time points, not multiple time points within the same culture, but they tested out multiple different time points. And here, what I'd like you to see is that there are significantly lower viral counts in all of the cultures all the way up to 3 hours post-infection. And of course, these times are relative, so they did account for giving time for the virus to actually enter the cells and to infect the HeLa cells. And so those 3 graphs that I showed you are the biggest ones that I wanted you to see in the paper, but if you'd like to look more, you're welcome to read that. But yeah, these results are really promising. And so that brings me what the future common cold cure will look like. So this cure targets human components, which is great. It minimizes the margin of resistance and mutations that could occur in viruses, and deactivation of an incorrect target virus is extremely unlikely as well. And because this EMP1088 is so specific, administering it to people would look like only 1 round of doses compared to multiple vaccinations like in the flu vaccine, where you have to become vaccinated every year. And not only is this cure going to be good for the common cold, but it can also help to lower the intensity of symptoms that are exacerbated by the rhinovirus, such as, again, asthma, COPD, and cystic fibrosis. And so with that, I will happily take any questions if anyone has any. I'm not a pro, but I'll try to answer.
In the graph that showed the different injection times and how that affected effectiveness of, or I guess suppression of NMT,
Yep.
does -1 represent an hour before the human cells were infected, or?
Yes, exactly. And so if you can see, the viral counts for -1, 0, and +1 are about the same.
Trina, I just had a quick question about the N-dimethylamine. Like, is there any specific reason that it targets specifically tyrosine? Like, does it also target other like aromatic amino acids? I think I might have missed it when you said it, but.
If you were to take the dimethylamine alone and to put it with other residues, then it probably would. But specifically this inhibitor and the way it interacts with the enzyme, it interacts specifically the tyrosine residue. Unfortunately, I don't have a picture of it to show you, but it's a very specific interaction.
Is the structure of the enzyme known? Like, has there been like cryo-EM done to like figure out kind of like what the enzyme structure looks like?
I believe so, but unfortunately, I'm not really well knowledged on that.
Okay, I was just wondering because
But,
like the structural bio. So I was just wondering because like the enzyme, like the active sites and all that is really cool.
yeah, I think they provide that actually in the supplemental information of the paper, but I
Okay.
can try to find more stuff if you're interested.
Do you think this form of treatment could be paralleled in other viral family groups, like the coronavirus family group?
So that's an interesting question, and I was wondering that. Although I don't have the knowledge to back up an answer, what I can say is that this process that it's targeting is known to be specific to the rhinovirus. And I don't know if the current coronavirus that we're dealing with has the same co-translational modification that's essential in forming capsid protein.
Wait, actually, I have a fun fact about the coronavirus. Okay.
Let's hear it.
If you guys remember the paper I presented on week 1 about my research, actually, instead of like this pathway that rhinovirus uses, coronavirus actually uses like kind of like the same RNA processing machinery like that the disease that I was working on also uses. So like in order to make like functional coronavirus proteins, they actually have to cut that intron out with an analog that's like similar to T-cell machinery. So like to the complex that I was studying.
That's so cool. Oh my God. This is the nerdiest conversation I've ever been a part of.
As soon as I said that, I was like, I can't believe I'm happy for this.
It's okay. We'll all bathe in the awkwardness together.
I was gonna say the scientist in Maya jumped out.
I'm such a nerd. I'm sorry.
But it's good you know that. That's actually an interesting fact.
It is.
Yeah, because maybe that's like a way to make like therapies towards it. It's like maybe towards like trying to degrade that enzyme or that protein complex or something like that.
Do you think this like this treatment or whatever could be used like do you think it would only be used for immunocompromised people or would they make it like available to everyone?
I really have no idea because it would be really expensive to administer this to everyone. And like I said, like everyone gets sick and get over it. And I don't know if it's a big enough issue to the inconvenience. I don't know if the inconvenience is big enough to spend money to get this for everyone. So they probably will just administer it to immunocompromised people.
So is this in like any trials now or is it just research right now?
So this paper specifically that I presented today was published in 2018. And when I read up a little more on it, I think they're close to beginning clinical trials. But I'll have to read up more on that to confirm.
Robbie, your question reminds me that like even if this was available to everyone to prevent against the common cold, it makes me wonder like how many people would actually like be motivated to get it, because I feel like there's so many people that don't take like the flu vaccine seriously already, or they're always like, oh, the flu vaccine's like a sham, it doesn't work, whatever. So how like the public would react to something like this. It's like, hey, you take this and you won't get the flu like ever, period, or something. I don't know.
Yeah, that is very interesting actually, because a lot of people, there's a growing anti-vax population.
Unfortunately, yeah.
Yeah.
Sort of looking at this from a different point of view, if this treatment did become publicly available, do you think conventional insurance would cover it, or would this procedure be unique enough, or like sort of non-essential enough to be considered something that insurance doesn't cover, so you have to pay out of pocket, or you need to get some sort of special, insurance that also covers symptoms like COPD and stuff before you try this treatment.
I feel like it definitely would be considered to be non-essential since so many people get sick with the cold and get over it. And so that's unfortunate for people who maybe don't even know they're immunocompromised, and then they'll become sick with maybe the rhinovirus and then they'll need it.
Yeah, I feel like insurance companies would be like, this is a pre-existing condition, we're not going to cover you, and they're going to drop you from your insurance or something like that.
Oh
Because
my
they do that all
God.
the time with like cancer patients and people suffering from other diseases that are really expensive. So
Mhm.
Also, would this treatment work if you didn't know you need it until you were infected with the rhinovirus? Would this be able to, like, if you already are experiencing cold symptoms, but it hasn't gotten to a level of severity yet where like your organs are shutting down, or you're paralyzed or something, could you inject this, and then since it's stopping this translational modification, would it stop further replication of the rhinovirus?
So preliminary results show that it actually happened based on this graph right here. And here they were, of course, the HeAL cells were infected with a rhinovirus strain for 6 hours. So the cells had time to replicate, the viruses had time to replicate, and then they dosed them with EB1088. And there was another figure that actually showed that the viral counts lowered in the paper. Where it was like multi-replication cycles. The ones that I showed you are single replication cycles.
I was wondering, could it put like a cap sort of on like how many replication cycles can occur before cells no longer have the translational modification occurring, and therefore the virus can't replicate?
Yeah, I have no idea about a cap. I would assume that there is a limit.
Trina, I think it's a really interesting article because, like, I think you're right. Like, at the beginning, you said the common cold or the common flu, a lot of people don't think twice about it. And then when you start talking about immunocompromised patients, it kind of reminded me of, like, kind of like right now, how people are kind of dealing with coronavirus. Like, because, like, you get the flu vaccine so that other people don't get sick, right? And that you don't get sick yourself. And it's kind of like coronavirus. Like, a lot of people, like, I went grocery shopping the other day and a lot of people weren't wearing masks. I was, like, one of the only people wearing a mask in the store. And it kind of makes me like worry about immunocompromised people because like we have the privilege to not take those preventative measures and stuff like that, but like what we're doing is not only impacting us, it's impacting like all of these people who's literally like their bodies can't afford to get sick like that. So I think this is really interesting.
Thank you, Myra. But yeah, I definitely agree. Like, I was in Target the other day, and my sister and I were the only people wearing masks besides the employees, and I was just like, that's disgusting.
Yeah.
I think it's really crazy. Colleges, like, I feel like they're not taking enough preventive measures. Like, they're just taking like fake preventative measures really to ensure people's safety for going back.
Yeah, how do you guys feel about the new measures that NC State announced yesterday?
I'm scared.
Me too.
Oh, yeah, I actually don't feel comfortable going back at all. I don't know if it's just me. Maybe I'm just paranoid, but I don't know. I don't want to go back.
How many of your classes have been moved online?
All.
Yeah, all of mine are online.
All of mine except for one have been moved.
Do you also go to state?
Yes. Wait, me?
Yes.
Oh, are there people here that don't go to state?
Yeah.
I don't go to state.
Oh, hey girl. I'm at NC State.
Yeah, Meredith is still, they haven't told us what they're doing, and they're saying that they want everything to be in person, and I'm just gonna take a gap year or something, because like that is so gross. Like, I'm gonna have to touch doorknobs and sit on desks, and I have I have like grandparents that I live with. Like, I can't, and they have like heart issues and lung issues and diabetes, so that's like all of the things. So it's like, seriously, like I don't know, like they opened our community pool and I was so mad. I sent like a really angry email because I was like, that is like the worst thing that you can open. There's like 600 houses in our community. Like, why would you do that? And they were like, we are like free to use the facilities. You don't have to. And I was like, oh, okay. Since we clearly don't have any sort of community like collective agreement for safety, it's ugh. I just think it's like, I've been looking at the graphs for I guess like the viral loads, because like I'm a public health major, and like we had all the information. Like since March, we've been looking at and malaria, and it's just like in other countries, even if it's continuing to happen, you can see like it's a bell curve. But for us, it's just like I don't think we're gonna get rid of this until like 2022 because places keep opening and shutting, and it's just very irritating because people are taking profit over people's lives.
I guess it kind of like the larger issue too in science is that we have all these, I think Dr. Hartstone touched on this last week too, that we have all these scientists who are doing all this research, but they like kind of like suck at saying it in like easy words for like the average person to understand, right? Like I feel like there's so much like misinformation. Like I saw this video on Twitter the other day of all these people like passionately supporting reopening, and like I don't know like what we can do as a society to ensure that people understand science like in an easier way, right? Because I feel like science is so complicated, and I feel like some of the scientists are just really bad at saying something in a way that's easy to understand for people who are not in the field. And I feel like that's where a lot of like the misinformation is coming from. I don't know.
I mean, I kind of agree with you. Wait, go ahead.
No, you should go.
Okay. I was just gonna say like our main problem isn't necessarily like scientists don't know what they're doing. I feel like part of the big reason is like the CDC was super vague about what to do in the beginning, but that's because like you said, the information, like we didn't have all the information. But the main issue is like the media and like a lot of the mechanisms in the country are so capitalist focused that like they just don't care about people's lives. Like it's all about stock market and like,
oh for sure,
you know, like it's like they just don't care if where people are gonna die because they want to go to Applebee's and they want to like, you know, go to the pool and they want their stocks to rise. And if, you know, people have to die for it, well, you know, that's just too bad.
Yeah, that's really true.
Oh, you got me at Applebee's.
Applebee's in my town open, and I'm like, why are y'all here? Y'all are not gonna be, y'all can eat this when we're all healthy. Like, why are y'all here right now?
Something that I think could help improve like citizen understanding of science is having more applied citizen science where, you know, scientists gather samples with like just normal everyday people, you know, normal citizens. And I think that would actually be very helpful in studies like this, because even if people don't trust science, a lot of people trust anecdotal data, stuff that they've experienced themselves, you know, even if they're not reading it statistically. So when they sit down in a lab with a scientist and just do some of the basic work, you know, with them, stuff that you don't really need specialized training for, they'll be more inclined to believe it because there's the whole I was there mentality. I was there when the flu was cured and stuff, you know. So you'll have like this societal push by a bunch of people who are like, yeah, I was there when the flu was cured, so I know for a fact that it's cured. Even if it isn't, even if that's not the entirety of the science, it would still move scientists into a better social standing and therefore like improve scientific messaging, I think. And also to play devil's advocate on reopening, I understand the whole, I do think lives are worth more than like capitalist endeavors, but at the same time, there is a real risk with coronavirus of like, if world economies start shutting down because the US and China have been closed for a significant period of time, which has stopped like something like 65 to 75% of the world's supply chain, another Great Depression style event could occur if that continues for like several months. And there is also a significant loss of life and like loss of value of life if something like that occurs. So I think that needs to be balanced out with like pure scientific human life comes before all, because like the human life loss from coronavirus eventually, right, might be equivalent to the life loss from a severe economic shutdown, unless there's like a significant systemic change in world economies towards like more social focuses, which I don't see happening in the near future.
Like I kind of, I mean, I understand your point, but the thing is, if we had just had a total shutdown for 2 months, like a proper total shutdown, we would have been done by now, you know, like we wouldn't have these issues. Like we're having more issues because we had such a half-hearted, completely like negligent response. Like, of course we don't want the economy shut down, but like if we don't have, like, you know, like in Italy, their partial shutdown was more than our total shutdown. And like we already know like the fact, like in Vietnam and in other countries, like they had like a really strong support system. So I think it all goes back to having like a really strong, like we don't have a system, like we don't have systems, we have like patchworks of like potential help for people, and we need to have like a proper healthcare system and a proper health education system that can like, you know, we can be prepared. And it's always not going to be a pandemic, but it's going to be something like next year. Like, you know, we always have some people dying from something, and we need to make sure that that doesn't happen. And we can reduce human casualties so much just by having everyone be able to access like a general practitioner, instead of just having like, you know, I don't even know what we invest in, like oil, like, yeah, okay, war, there you go, oil and war. Yeah, but yeah, that like, that's all we invest in. And if we can, I mean, it's obviously not easy, but if we can do it on a local level and maybe move up from there, I think it's worth it. And also check out the Poor People campaign. They're actually looking for people who are interested in science to like work with them, because it's like, it's the program that Martin Luther King Jr. was working on when he was assassinated, and it talks about healthcare for all, and it's like they have a moral budget, I think that's what's called. So yeah, like it's all connected.
Wait, Homa, could you send us an email like about the last thing you mentioned so we can post it on our blog as well, so other people.
Yes, I will do that.
Yeah, I definitely would love to check that out. Wow,
okay, I just made a
I'm
note.
pleasantly surprised. I wasn't expecting so much discussion to come with my paper. I was worried it would be boring.
No, it was honestly such an interesting paper.
Oh, yay.
And it was easy to understand, which is nice.
I'm happy. I tried to break it down in the way that like I had to break it down for myself, because I'm not a microbiome person at all, and I barely just took biochemistry, so this is certainly out of my comfort zone.
I think you did a great job explaining the paper, though. Considering it is out of your comfort zone, that was pretty awesome.
Yeah, Trina, you and John did an amazing job.
We will definitely be posting blog posts on both John and Trina's work, so you guys can definitely like hop on the blog and just post a little bit about what we talked about. I think the vaccinations, like being anti-vax or like pro-vax. Obviously, most people here are probably pro-vax, or just like COVID related, what you guys think should be, you know, implemented as safety precautions for colleges, for the general public. I think that would be a great thing to discuss on there. So hopefully we can have that up in the next couple of days, and we will send you guys an email, and then we can continue the discussion there.
Yeah, thank you guys so much for joining this week. And just a quick reminder to please sign up for dates. Week 8 and week 9 near the end of July, like July 20th to July 31st. We have a couple of open spots. So, and I really encourage all of you to go and try to present. It's a really great way to break down complex information into something simple, and it'll really help you when classes start back up and when you go into these research positions. Thank you guys for coming.